The keynote lecturer is my dear friend Pierre Magistretti. And Pierre, as most of you know, has had an illustrious career. Only a few people can have two full-time positions at the same time and look as good as Pierre. So Pierre is spending some of his time in Saudi Arabia, where he's having a chance to oversee the development, I think, of sort of a rebirth of biological sciences there, and of course is at the EPFL in Lausanne. So Pierre is going to talk to us about neuron-glia metabolic coupling, relevance for neuronal plasticity and functional brain imaging. Pierre.
Thank you very much, Bruce, and thanks to the organizers for inviting me for this lecture and giving me the opportunity to present a long story in a way that started several years ago, almost 3 decades ago, when I was a graduate student, and that's when I became interested in astrocytes. And I will bring you through the most recent work, which is connected to this older work. So my main interest over the last few decades has been to try to understand the mechanisms that underlie the coupling between synaptic activity and the electrochemical events that occur at the synapse and metabolic and vascular responses. We particularly focused on metabolic coupling. And this is a topic that is of general interest, I think, for physiology, to understand how synaptic activity is coupled to energy metabolism. It was already demonstrated at the end of the 19th century by Sherrington, and then later with Lou Sokolov using the 2-deoxyglucose technique, also for metabolic aspects. But another dimension of this coupling is, of course, the fact that actually functional brain imaging techniques detect signals that are related, hopefully, but actually that's pretty sure, to these electrochemical events. So understanding how the coupling occurs is also of interest in this dimension. Now, in general, up to a few years ago, the idea was that this coupling was pretty straightforward, with active neuron releasing some vasodilatory substances which would increase local blood flow and provide energy substrate to the active neurons. And some of it may still be the case, but actually there was a key player that was missing in this picture, and these are the astrocytes. We are here at the Glia meeting, so I don't have to spend too much time to convince you that these cells are important. These are 2 drawings by Santiago Ramon y Cajal, who already identified and characterized these cells. This is a more modern view using GFAP as a stain. And you can appreciate that Cajal already identified that there are contacts between these astrocytes and blood vessels, but not much more than that in terms of coupling. But more recent work, neuroanatomical, at the EM level, at the immunocytochemical level, has shown that actually astrocytes possess processes that ensheath synapses. This is not complete. It's actually quite dynamic. But you have in this 3D reconstruction of EM image by Graham Knott at EPFL, you can see here a presynaptic terminal, a postsynaptic element, and in blue are these astrocytic lamellae that are around the synapse. And then the other processes of astrocytes actually surround intraparenchymal blood vessels, as shown here in negative from in this image. So in a more artistic view, one can see and appreciate that actually astrocytes are interposed between neurons, and in particular they have these lamellae that surround synapses and other processes around the capillaries. And more importantly, from a functional point of view, that at the level of these processes, astrocytes express receptors for a variety of neurotransmitters, and also in particular reuptake sites, in particular for glutamate, which is, as you know, released at least 80% of the synapses. So they can sense synaptic activity, in particular glutamatergic. And at the level of the end feet, these processes that surround the capillaries, they express transporters for a variety of substrates, including glucose, of course, and also lactate and amino acids. So, as I said, we studied the mechanism that would allow this coupling, and as I mentioned earlier, many years ago, I became interested almost by chance in astrocytes and showing that they actually were involved or are involved in such a coupling. And over the years, we described two main mechanisms of coupling between neuronal signals and energy metabolism that actually put astrocytes at the core of this coupling. And the first mechanism that we described was here in 1981. I show this picture because here I am a student doing my PhD in shorts and a beard. And since there are many students here, that's when I was at the Salk Institute in San Diego, and chose one example. I don't say that's the only one, but an example of a work that has been carried over, in this case, over 30 years. And what I found then, and that was the core of my PhD thesis, was that a peptide that has been discovered very recently, that was a time when peptides in the brain were discovered almost by the week, that VIP promoted an unexpected effect. Was supposed to look at excitability, but for some reasons which I don't have time to go into, mostly by ignorance, I tried to see whether VIP had an effect on glycogen levels. And I found that actually VIP promoted glycogenolysis, so that gave the idea, the first hint that there was a signal released by neurons, VIP, that could act on astrocytes to provide energy substrates. And how could I say that it was an interaction with astrocytes? Well, the reason is that glycogen, is essentially localized only in astrocytes. You have here a capillary, with the capillary wall, the endothelial cell. Here you have one of those astrocytic end feet that I showed earlier, and here are these electron-dense granules, which are actually glycogen granules. And here you have the neuropil with synapses and pre- and postsynaptic elements. And in fact, I think this was one of the first demonstrations that a neuronal signal could exert a physiological action on astrocytes. We found out that actually only a restricted set of transmitters is glycogenolytic, in addition to VIP, analog PACAP, and then noradrenaline, mostly via beta-adrenergic receptor and the cyclic AMP cascade. And later on, we found that actually also adenosine and ATP can promote glycogenolysis. So at the time, the conclusion was there were some transmitters, noradrenaline, VIP, adenosine later, that would mobilize glycogen from astrocytes, promote glycolysis, and provide energy supplies, additional energy supplies to neurons. And since at the time I was also characterizing the morphology of the VIP neurons, which actually belong to a very homogeneous population of intracortical neurons, bipolar neurons, which essentially define cortical columns. I proposed at the end of my thesis that actually these neurons could be some sort of neurometabolic transducer because they are activated by specific afferents to the cortex and then translate this input into a metabolic response to mobilize energy within cortical columns. And this was to be contrasted with the noradrenergic system, which, as you probably know, is organized in a completely different manner, actually tangential to the VIP neurons, meaning that noradrenergic fibers can actually somehow prime the whole cortex simultaneously across functionally distinct cortical areas. And later on, Ray Swanson and colleagues showed that a sensory stimulation, the whisker-to-barrel pathway, was indeed decreasing the density of glycogen granules in the somatosensory cortex, showing that a physiological stimulation would actually mobilize energy in the relevant cortical area. Then the second mechanism for coupling that we described came about 10 years later, and that was work that was done with Luc Pellerin, who was a postdoc in my lab after his PhD at Montreal. And we showed that glutamate uptake, a well-known at the time already function of astrocytes, was stimulating aerobic glycolysis. So what is aerobic glycolysis? Actually, it is the production of lactate in the presence of oxygen. And this is a well-known metabolic pathways characteristic, by the way, of tumoral cells. It's called the Warburg effect. And it's been now re-evaluated in terms of its relevance in tumoral growth and plasticity. And this aerobic glycolysis means that glutamate could stimulate glucose uptake and the production of lactate by astrocytes. And we proposed that this was another mechanism to couple neuronal activity to glucose utilization. Of course, in a way much more relevant or with a greater impact than glycogenolysis evoked by VIP or noradrenaline, because as I said, about 80% of the synapses release glutamate. And this slide summarizes several years of work by postdocs and graduate students, where the mechanism for this glycolysis elicited by glutamate is as follows. Glutamate being released by neurons and acting, of course, on postsynaptic neurons to exert excitation. Glutamate is taken up by astrocytes using the electrochemical gradient of sodium with a stoichiometry of 1 glutamate with 3 sodium ions. Now, this uptake actually puts a metabolic burden on astrocytes with the conversion of glutamate to glutamine for its recycling and reestablishing, at least in part, the presynaptic glutamate pool, not 100%. Some glutamate can actually also be used as an energy substrate. And this costs 1 ATP. And then for 3 sodium ions to be extruded by the pump, that will also cost 1 ATP. And we showed that it's actually a mobilization of the alpha-2 subunit that is involved in this. Now, this burden results in a decrease in ATP. We showed also this with Jean-Yves Chaton, meaning that, like in any cell, glucose uptake is stimulated because of disinhibition of hexokinase and phosphofructokinase, and this leads to glucose uptake. And as I said, the surprise was at the time that lactate was the substrate that was released. Doesn't mean that glucose is not used by neurons. Actually, neurons have GLUT3 transporters, and, but they mostly use, and this is work by several labs, by Moncada, Bolanos, and others, in the pentose phosphate shunt to produce reducing equivalents in the form of NADPH. So this mechanism provides a pretty straightforward mode to couple synaptic activity to glucose uptake into the brain parenchyma and the provision of an energy substrate, lactate, for neurons. So this gave rise to what is now known the astrocyte neuron lactate shuttle, whereby a neuronal signal, glutamate, through specific transporters is taken up, disrupts sodium homeostasis. This costs some energy. Glucose is taken up. We showed later that actually astrocytes contain the LDH5 form, the muscle form, which is enriched in tissues that produce lactate, that neurons only express LDH1, the heart form, which is enriched in tissues like the heart, which use lactate as a substrate. We also characterize the transporters that allow this transport of lactate from astrocytes to neurons, with the 1 and 4 form, in particular the form subtype being astrocytic, and 2 for neurons. And from a metabolic point of view, the view that emerges is that astrocytes are mainly glycolytic, even though they express mitochondria, of course. But as I will show you in the next slide, there are some, the expression of particular enzyme makes them glycolytic, whereas neurons are mostly oxidative. Now, indeed, neurons and astrocytes have complementary metabolic profiles, with astrocytes containing glycogen and the enzymes for glycogen. they express this form of this enzyme, PFKFB3, which produces fructose 2,6-bisphosphate, which is a positive modulator of glycolysis. They express this form of pyruvate kinase, the M2 form, which is enriched in tumoral tissues, for example, which produce lactate, so which is enriched in glycolytic tissues. And the PDH, the enzyme that allows the entry of lactate, of pyruvate and lactate, once converted to pyruvate, into the Krebs cycle, is actually close to saturation at basal conditions, because it's highly phosphorylated. Neurons, on the contrary, they don't express glycogen. Actually, the enzyme glycogen synthase is constantly degraded by a proteasome process. Same for PFKFB3. It's also expressed but constantly degraded by proteasome processes, meaning that actually neurons cannot upregulate glycolysis. They express the form of pyruvate kinase, the M1 form, which is enriched in oxidative tissues, and they have a high activity of pyruvate dehydrogenase. This has been summarized in a recent review that we wrote for Neuron. So in summary, glycogenolysis occurs only in astrocytes. Glycolysis occurs predominantly in astrocytes. Both pathways produce lactate. Now, glutamate is not the only signal, extracellular signal that promotes glycolysis. In fact, both Felipe Barros and Brian McVicker here have shown that extracellular potassium, which is a feature of activation, whenever neurons depolarize, they release glutamate, and they also lead to an increase in extracellular potassium between anywhere to 5 to 10 millimolar. And this increase in potassium, by modulating pH in the alkaline direction through different mechanisms, promotes glycolysis and actually even glycogenolysis, as shown by Brian. So there seems to be redundant mechanisms, but probably because it's very important for neurons to receive adequate energy supplies. Now, since we had shown that going back to glutamate, since glutamate seems to be important for the import of glucose into the brain parenchyma, we did a series of experiments where we actually either down-regulated the expression of the glial glutamate transporters, and keep in mind that 90%, if not more, of glutamate removal from the synapse is operated by astrocytes, only minimally by presynaptic terminals. So the idea is that if this is down-regulated, or if we could use knockout mice for this transporter, then stimulation of a sensory pathway, for example, would impair glucose uptake because of this tight coupling that we showed. So we tested this in collaboration with Gilles Bonventeau and Egbert Welker, using this well-established sensory pathway, the whisker-to-barrel field. One can stimulate selectively a vibrissa and look in the corresponding barrel field in layer 4 of somatosensory cortex, an increase in 2-deoxyglucose signal by autoradiography, an imaging signal, if you wish, of glucose utilization. Now, we used two knockout mice for GLAST and GLT1, which are the glial glutamate transporters. And as you can see here, this is the control, the wild type, and this is the knockout for GLAST, and this is the knockout for GLT1. And you see that actually the signal for 2-deoxyglucose uptake following stimulation of the vibrissae is not seen. And the same when we used a oligonucleotide antisense to down-regulate the expression of GLT1. You have here the control, the contralateral side stimulated, and you see a nice image here of 2-deoxyglucose autoradiography. But here, and the control obviously has no signal, but here where we injected here the oligonucleotide antisense for GLT1, you see the stimulation of the vibrissae markedly decreased the imaging signal, whereas on the control side, we still see the 2-deoxyglucose autoradiographic signal. So this has an implication for the interpretation of the imaging signals, certainly for the PET signal for 2-deoxyglucose, F18FDG. It doesn't question the validity of the technique because the initial signal is glutamate, but clearly if alterations in this signal are seen, it may well be that it's due to, for example, a loss of glutamatergic synapses, like the case, for example, in Alzheimer's disease, and indeed there is a massive decrease in the 2DG signal, but also some abnormalities could be present in these coupling mechanisms, pointing at possibly mechanisms of pathology located in astrocytes. As to the fMRI bold signal, there is also this transient glycolysis may be relevant for the production of this signal, because the idea is that whenever there is activation, glucose is taken up, but not immediately oxidized in astrocytes, or only marginally. It is released as lactate, and only then lactate is oxidized after transformation to pyruvate. And what happens with fMRI? Well, first some data obtained by Mark Rakel already in the late 80s, but confirmed even more recently, that during intense visual stimulation, using PET now, I go back to PET for a second, there is an increase in blood flow, there is an increase in glucose utilization, but the increase in oxygen consumption is actually much, much smaller, not commensurate. So meaning that at least for a certain time window, there is an uncoupling between glucose utilization and oxygen consumption, meaning glycolysis, aerobic glycolysis. Now, this means that during activation, this is baseline, you have in red oxyhemoglobin, in blue deoxyhemoglobin, mostly on the venous side. During activation, as I said, there is this increase in blood flow, meaning an increase in the delivery of oxyhemoglobin, but because there is not an immediate use of glucose oxidatively, quite paradoxically, there is an increase in oxyhemoglobin, or an increase in the ratio between oxy and deoxyhemoglobin in the activated area. And this is what actually provided the impetus for Seiji Ogawa to develop bold blood oxygen dependent level imaging, because the idea was that actually there should be being more the ratio between oxy and deoxyhemoglobin being changed, the paramagnetic properties of the tissue would change in the activated area and provide a signal which is now very broadly used. So in summary, the fact that there is a transient non-oxidative use of glucose during activation provides a time window that is consistent with the increased oxyhemoglobin in the activated area and the bold signal. Now let me move to the second part of my talk, which deals with recent work we did on the topic that we call metabolic plasticity. The question that we asked is the metabolic coupling between astrocytes and neurons subject to plasticity? A lot is known, of course, in terms of synaptic plasticity, in terms of gene expression, of cytoarchitectonic rearrangements, of increased release, increased receptor expression. The question is, are these synaptic changes that occur during plasticity somehow associated with a plasticity of this neurometabolic coupling, or even is this neurometabolic coupling necessary for plasticity? So we engaged 4 or 5 years ago in a quite time-consuming set of experiments whereby we subjected mice and rats to learning paradigms, two main ones. One is spatial learning using the 8-arm radial maze. As you probably know, there are 8 arms, 3 are baited with food, and within a few days, 3 to 4 days, the rodents are pretty able to go straight to the baited arms by using spatial cues. And this is a one-trial learning, very solid and reproducible very easily, and that's inhibitory avoidance. So mice or rats are placed in this 2-compartment cage. One is lit, the other is dark. As you know, rodents like to go in the dark, so they move very quickly in the dark compartment. The day of experiment, when they move into the dark compartment, you give a small electric shock, and then the next day you test them, and they remember that something fishy is going on in the dark side, so it takes a long time for them eventually to get into the dark side, And so latency means that they have learned. If they have learned, they have a long latency. Now, the idea was that at the end of these learning experiments, we would monitor which areas of the brain have been activated differentially between control and learning using the 2-deoxyglucose technique. And then, because that indicated that somehow these regions were engaged in the learning, and then we used laser microdissection to microdissect these activated areas, and then looked at gene expression, in particular of genes that are involved in this neurometabolic coupling. And as I said, this involved work by several students and postdocs, and I won't go into detail, but what really was interesting is to see that essentially all genes involved in glycogen metabolism were upregulated by learning. So this brought me back to the time I was in shorts in the lab, and I thought, well, maybe let's try to block glycogenolysis during learning. This was done with using this compound, which, by the way, is an anti-diabetic compound that blocks glycogen phosphorylase, or to downregulate the expression of the transporters of lactate during learning. And these experiments were done in collaboration with Cristina Alberini in New York, who has extensive experience with this inhibitory avoidance learning and the molecular analysis of this learning. And so the experiment was pretty straightforward, inhibitory avoidance paradigm. 15 minutes before training, we injected bilaterally in the hippocampus DAB. We tested for short-term memory within 1 hour or long-term memory, 24 hours. And the idea was either to block glycogen phosphorylase and prevent the formation of lactate, or to prevent the export of lactate between astrocytes and neurons, using oligonucleotides against the specific transporters. So this led to this paper published in 2011, where we showed that indeed astrocyte to neuron lactate transport is required for long-term memory, something that was a bit of a shock in a way, or certainly unexpected for the, let's call it the neuronal community, because in a way a cell type, which is thought for a long time to be just glue, the astrocyte, was releasing a compound that was for a long time thought being as the metabolic end product, lactate, and that this was necessary for long-term memory. And here are the data, very briefly. I just show a few of them as this has been published, but essentially after a vehicle injection, you see that the latency to get into the dark compartment is quite long compared to control. If DAB was injected, the latency was much, much shorter. And this was observed only at 24 hours. There was no difference when the animals were tested at 1 hour. But more strikingly is that when we co-injected lactate with the DAB, we actually rescued the memory, because animals actually, again, had a long latency to get into the dark compartment. And this lactate was definitely coming in the experiment from glycogen, because this is a microdialysis analysis of the lactate present in the extracellular fluid during training, and here is in the presence of DAB, meaning from glycogen, meaning astrocytes. And then finally, again, this result may be of interest, the cellular counterpart of learning and memory, at least one of them, is the long-term potentiation. And in vivo, in the hippocampus, we could show that indeed LTP could be induced both in the control animals and in the DAB injected ones, but it was maintained only in the control ones. DAB actually inhibited the maintenance of LTP. So in summary, I didn't have time to show you this, but again, it's published. If we block if we downregulated MCT1 or 4, we lost memory, and this was rescued by lactate. If we downregulated MCT2, also memory was lost, but lactate did not rescue, and which is pretty straightforward because lactate could not enter into the neurons. So in summary, lactate is released with inhibitory avoidance training in the hippocampus. Blocking glycogenolysis blocks both memory retention and lactate release, as well as the molecular changes known to underlie long-term plasticity and memory formation and LTP. The conclusion is that transport of lactate from astrocytes to neurons is required for memory consolidation, and somehow that glycogenolysis and astrocyte neuron lactate shuttling are required for long-term memory formation. But at the end of these experiments, we had a puzzling question. Actually, this is taken from the paper. We actually, when we used glucose to rescue the DAB effect, the rescue was only marginal. It took longer, and it was much, much smaller. So we suddenly started to think that maybe lactate was providing more than energy, maybe energy for all the processes linked to plasticity, such as cytoskeletal rearrangements or gene induction, or translation more precisely. But then that maybe lactate could be a signal for plasticity. So we went back in vitro and used primary neuronal cultures to see whether lactate and other energy substrates could modulate the expression of genes that are known to be involved in plasticity, such as, for example, ARC, BDNF of ZIF268. And we were actually quite surprised, but actually at the same time pleased, to see that actually L-lactate was the only one of these substrates to be able to induce ARC and ZIF, and also CFOS, which is more of a marker of activity. D-lactate, the inactive isomer, didn't do anything. Glucose was inactive, and pyruvate was also inactive. And actually, at 4 hours, these experiments on ARC and ZIF were at 1 hour. At 4 hours, BDNF was also induced. And this, after a long search, we tried to understand what was the mechanism for this effect of lactate, specific for lactate, on plasticity. And probably we should have thought of this much earlier, but this is about the last pathway that we tested, was actually that lactate is a positive modulator of NMDA transmission. Lactate promotes plasticity gene expression by potentiating NMDA signaling in neurons. This was published a few months ago. And very briefly, you can see that the effect of lactate is blocked by MK801, and it is also blocked by inhibitors of the MAP kinase ERK1/2 cascade. Here you see that the proteins for these genes are actually are blocked in the presence of lactate. And lactate indeed amplifies the NMDA current, the glutamate NMDA current. Here is the control. Here is in the presence of lactate, and the increase in calcium that is evoked by this activation of the NMDA receptor. So in summary, lactate stimulates in a time and concentration-dependent manner the expression of plasticity-related genes, ARC, ZIF, to a lesser extent CFOS, and BDNF in cultures. We also did intracortical injections of L-lactate and found the same. This effect is mediated by NMDA receptor activation. It is blocked by MK801, by blockers of the glycine site, and it involves the ERK1/2 MAP kinase signaling pathway. And then lactate potentiates glutamate-evoked currents and increases in intracellular calcium. And we think that it's a change in the ratio between NADH and NAD that mediates the effect of lactate, because when lactate is transformed to pyruvate, NADH is formed. So in summary, lactate acts as a signaling molecule and not only as an energy substrate. Now, very quickly, the last couple of slides showing very recent data. In these experiments, we used what we would call a candidate gene approach. We looked at genes that we thought would be involved in plasticity. But now we have engaged in an mRNA sequencing approach or transcriptome analysis. And in fact, we found even more. Now we are about up to 60 genes that are induced by lactate in a selective way in neurons. And many of these genes are involved in plasticity. Some are unknown. So it's really a Pandora box that we are actually opening. And then a question that we asked very recently is, well, if glycogen is so important for plasticity, it would be interesting to see where these glycogen granules are within the astrocyte. Are they close to the cell body? Are they close to the synapse? And so what we did was to use actually a very unique, I think, device or experimental approach that involves the construction of a 3D virtual reality based on a reconstruction of EM sections. So the way we did this was, let me show you very briefly. This is the standard EM block, and standard sections can be prepared with milling EM. And then segmentation is performed. You can see here a blood vessel. You can see in green here, maybe you can guess, these are astrocytes. On each section, these profiles are segmented. In gray are the axons. In blue are dendrites. And then you actually take this information, this segmentation information, and use appropriate software to put this information in a 3D full immersion virtual reality. And you see here in this cave, as it is called, the blood vessels in red, and then you start walking. You take a dip into this block. Here in green are the astrocytes, in purple the glycogen granules. Here you see a dendritic process and the synapse. And you can actually use this. Of course, it's kind of exciting to do this, but you can use it quantitatively to see actually where these glycogen granules are localized. And again, you have here, for example, an astrocytic process in a section with the glycogen granules. And here you have the 3D reconstruction. And what we found is that these glycogen granules are organized in clusters. And it turns out that at least half of the clusters are around, in astrocytic processes, around presynaptic boutons. Some are on spines, some are near the endothelial cells, which is not surprising because that's where glucose comes. And then, here, sorry. So this is a work in progress, but of course we would like to see now during learning if there is a redistribution or a change. So let me conclude by saying that, so we started with this almost serendipitous observation, finding that VIP is glycogenolytic. We then found out that other few transmitters can do it. This leads to lactate production. Same for glutamate through the uptake and then the mechanism I described. And both pathway converge to produce lactate. And I showed you that actually this lactate may well be an energy substrate converted when it is converted from lactate to pyruvate, but it is also a signal that by modulating positively the NMDA receptor turns on plasticity genes. And more recently, we have actually evidence that, and we're not the only ones, that lactate in the presence of oxygen always actually has some neuroprotective effect. Here, in a collaboration with Lorenz Hirt, who has developed, who has established the middle cerebral artery occlusion protocol, we showed that intracerebral ventricular, but even IV administration of L-lactate up to 1 hour after reperfusion decreases the infarct size and improves neurological recovery. So that is a kind of an interesting finding. And in a collaboration with Jeff Rothstein, actually, Jeff focused considerably on the role of oligodendroglia in ALS. And what was shown that, in fact, there is a decreased expression of MCT1 transporters in in general, in the SOD mice, not in motor neurons, is a mistake, and in pyramidal cells of ALS patients. And then disrupting MCT1 transporter produces damage and neuronal loss. So the idea is that a transfer of lactate from oligodendrocytes to neurons is damageable, is important for axonal function. And actually, Klaus Nave's lab has published a series of very nice papers showing that indeed the delivery of lactate from oligos to axons is important. And the idea is that this lactate can originate from astrocytes, and that because of astrocytes and oligos are coupled through gap junctions, this is the way to deliver the lactate at the right place on the axons. So in conclusion, starting with this question, we expanded and described two mechanisms for this coupling, and then also their implications for functional imaging and plasticity. And essentially, this observation indicate that a few neuronal signals can signal to astrocytes to produce during activity lactate, which has several positive functions in neurons. And of course, the challenge now is to understand exactly what is the relative contribution of each one of these signals in space. We have some ideas when you compare VIP to noradrenaline, and in time to provide this extra lactate during activation. Just to mention the collaborators, Igor Alamand has been in my lab for many years and has been a very strong right-hand man for all the work. Jing Yang, Pascal and Evelyn and Jean-Marie have worked on the most recent paper on lactate and NMDA. Monica, Giulia, Gabriele, Silva and Fulvio have done work on the behavior. The inhibitory avoidance work was done with Christine Alberini. The ALS work was done in collaboration with Jeff Rothstein and Brett Morrison. I mentioned the work on MCAO and Lawrence Hirt. And more recent work with transcriptome analysis and 3D visualization was done at KAUST by 3 postdocs there, Hubert, Corrado, and Heike. Thank you for your attention.
So, very nice, Pierre. I expect that there'll be some questions, and while people rush up to the microphones, let me ask a couple of questions. One that it's really fascinating, this new observation about lactate having an impact on NMDA receptor function. How long does that take? I mean, is it, can I bathe a synaptic, rather a brain slice in lactate and suddenly see, enhanced NMDA, or does it require induction of new protein or receptors?
No.
What's the mechanism?
No, this it's a pretty rapid effect, the amplification of the NMDA current. You see here, it's a matter of seconds to minutes, so it's a rapid effect.
So you put it on, and then minutes later,
Yep.
it's enhanced.
Exactly. At least this in the experimental condition. Now in situ, we would have to look at it.
But I thought it was. I thought there was a second messenger, but no gene expression involved.
No, not on this one. It's actually the NMDA is known to activate the ER kinase and MAP kinase pathway and lead to gene expression increases. So all that lactate is doing is really making the NMDA receptor signal more efficient in a way.
Okay. I have some other things, but Brian.
Okay, great talk. I love that a little bit with the this way to visualize the glycogen around the synapse. Very impressive, this cave, but it's quite intriguing that such a large percentage is at the synaptic regions and the astrocyte around the synaptic regions. Do you think this reflects a certain metabolic demand that the astrocytes are supplying and needed but unmet energy need of the synapse? It's kind of intriguing that there is this energy stored in the astrocyte so close to an area of high metabolic demand.
Yeah, I mean the problem, why design something more complex? I mean, why have glycogen there instead of having it at the synapse? Well, there is a series of nice work by Juan Guinovar and Bolanos and Moncada. They showed that actually, I mentioned it in passing, neurons express glycogen synthase, but it is constantly degraded by a proteasome process. And if this degradation is inhibited, in other words, if glycogen can accumulate in neurons, then actually neurons die. I mean, they have an apoptotic effect on neurons. And in fact, there is and I think Bruce must know it, is juvenile myoclonus epilepsy, which is also known as Lafora body disease. And Lafora bodies are nothing but glycogen accumulation in neurons. And the reason of the disease is that there is a mutation in this process that leads to a disruption of the process of degrading glycogen synthase as it is formed. So somehow neurons cannot afford to have glycogen.
And they can't break it down, right?
And they cannot break it down
That's
anyway.
the problem.
That's the problem.
Please.
So thank you very much. So in the CNS, there are quite a few cells other than astrocyte and neurons. What about their metabolic capacities? For example, microglia and oligodendrocytes, OPCs.
Yeah, this is something that would be worth studying. I don't know how, I think there are some studies out there in the energetic demands of microglia. I think it's a very interesting topic. I don't know much about it, but it would be, I think it's something we always had in mind. One day we should look at that in the lab. It's a very good question.
I have another question.
Yeah.
Regarding the role of lactate, it seems that it's very robust. Do you think that it acts directly works on glutamate receptor, or it actually through some other receptors which binds to the NMDA receptor?
So what we are exploring now, because we have this hint that somehow this effect of lactate on NMDA signaling is due to a change in the NADH-NAD ratio. So in the change in the redox environment. Now, as you probably know, the NMDA receptor has several redox-sensitive sites to cysteines. And so what we're doing now is to try to express the different NMDA subunits in HEC cells, and then some mutated for these redox-sensitive sites and see whether we lose the effect of lactate. So in a way, I'm just guessing, but just to answer your question, if there would be other receptors that have these redox-sensitive sites, who knows, maybe lactate could modulate their activity.
So may I ask another question?
We're going to give you 3 for 1 today. This is a special one-time offer.
Okay, thank you very much. So if the NADPH, the ratio is very important, then would activity change of LDH may actually regulate the NMDA receptor mediated signaling? Thank you.
In fact, it's not completely related to your question, but maybe you're aware there is a paper that was recently published in Science by the group of Inoue, not the one here, but another one, who showed that actually blocking the conversion of lactate to pyruvate by blocking LDH with oxamate in neurons, but also injected in astrocytes, so preventing the release of lactate, actually has an anti-epileptic effect. And they developed now a new experimental anti-epileptic drug based on the inhibition of LDH. It's a Science paper, came out a month ago or so.
So, Bill.
I'm referring to my notes here because I'm not good at this. So near the synapse, provision of lactate to the neuron compensates for energy use, and that's gauged by uptake of glutamate by the astrocytes. There's a linkage between glutamate uptake and lactate provision.
Yes, exactly.
Now, in the white matter, does a similar thing happen? I mean, how does the astrocyte, even if oligodendrocytes provide the lactate into the axon, they're getting it from the astrocyte. How does the astrocyte link the energy demands of the axon to the production of lactate in the white matter?
Yeah. So, as I said, Klaus Nave looked, and his group, they looked at this the necessity of aerobic glycolysis for the maintenance of axonal integrity, let's call it like this. And the idea there is that lactate can be formed in astrocytes and conveyed through oligos, through gap junctions to the axon. Now, what is the initial signal? I don't know, maybe Bruce has an answer. It could be potassium. I mean, I think Brian and Felipe Barros showed that potassium can promote also glycolysis. So given the restricted environment in the, around the axon, maybe that could be the signal.
So, Pierre, I had one. So the baseline level of lactate is extraordinarily high in the brain. It probably sits in the central nervous system, not the peripheral nervous system, close to a half a millimole.
Yes.
So
No. Yes, half a millimole. Yes,
yeah,
yes,
so that's a lot of lactate. It's tough to see signal and noise. Do you have any thoughts about that? This is not a conventional pharmacology for sure.
So what I personally think, and I think there are more people call it names that they like volume transmission or something that is non-synaptic, in other words. My view is that actually lactate can locally provide a homeostatic signal for, first of all, energy demands, and also for plasticity. It's not a point-to-point signal like glutamate or even the peptide transmitters or GABA. I mean, it's probably something that is more in the neurohormonal type of regulation. As I said, some people call it volume transmission. It's probably something that goes beyond the synapse and engages a cortical volume, but in important ways, I would think.
Right.
So have you seen any evidence for the new lactate receptor, that's a G protein coupled receptor, or GPR81? Have you seen any evidence that this might be involved in some of the processes you're looking at?
So we tried, of course, this is one, as I told you, before we got to the NMDA signaling, we tried a lot of things, and we couldn't see anything. And of course, we looked at the GPR81, I think it's called, it's lactate receptor, identified actually in adipose tissue as a negative modulator of cyclic AMP formation. It's coupled to GI. And there is actually this receptor in the brain. The thing is that this receptor is non-stereospecific for lactate. DNL, sorry. So our effects are extremely stereospecific. And the other point is that when we use, and one has to be careful, but these lactate transporter inhibitors, we didn't do the downregulation, but the pharmacology, we find that at least 2 out of 3 of these transport inhibitors inhibit the effect of lactate. So somehow lactate has to enter the neuron, be transformed to pyruvate, produce NADH, change the redox environment, and modulate the effect, the efficiency of the NMDA receptor.
So, Bill, are you just stretching, or do you have something important to say? Please, Bill, we have to hear you.
In going back to axon tracts, oligodendrocyte precursors are known to have glutamatergic synapses with axons and to receive synaptic input. Is it possible, can you think of some scenario where astrocytes are in contact or close to those synapses in axon tracts, and that is the glutamate spillover from those synapses is somehow the trigger for lactate production and transfer either directly into the unmyelinated part of the axon or into myelinated axon via oligodendrocytes?
Yeah, that's a good idea. I don't know. This is my answer, but I think it's interesting. It's a very good idea. Yeah.
So let's thank Pierre for a really broad and, oh, wait a minute, we have one more person. He will be our last question asker.
So, I'm sorry. There's one thing it's hard for me to understand. The timing issue. So the lactate is released from astrocytes, I guess, takes in the range of seconds, I guess. The astrocyte activity occur also in the range of 20 seconds, I guess. So the neurotransmission occur in the range of milliseconds. How do you comment on this? For me, it's not so easy to understand. Yeah.
I think one has 3 types of time constants. One is the, let's call it the electrical time constant, or the exchange of charges across the membrane that generate EPSPs, or action potential, or EPSP. This is within milliseconds. Then you have the metabolic time frame, which then takes seconds to have glucose being imported and lactate produced. And that lactate produced can modulate the activity of an NMDA receptors, but then the consequence of this is within actually an hour or even more, because you turn on a gene expression program. So I think one has to dissociate the three time courses at least. A very fast one, which is the signaling, a second one, which is the metabolic, and the third one, which is the gene expression.
Thanks.
Okay, well, Pierre, thank you very much. You stimulated the group, and we appreciated your lecture very much.
Thank you.